Hi, everyone. Welcome to our webinar. Thank you very much for joining this session. I'm Krishna Pulusu. I'm VP of Product Innovation and Strategy at Turbine, and I'm based in Cambridge, UK, where it's swelteringly hot today. And have my colleague and head of targets, Csilla Hegedűs, who's based in Budapest, Hungary. So I've spent 10 years in the pharma industry, and the question and the topic that we're going to discuss today is not going to be a surprise to any of you. So the target discovery to validation journey is not only complex, but also sets up the probability of success for everything that happens downstream in the drug discovery and development pipeline. And accelerating this journey is not only important to get the right drugs, better drugs to patients faster, but also make sure that we are understanding and modeling biology, disease biology, in the right way possible. And that's going to be our topic for today. Turbine, we are a virtual cell company, and in a second, I'll tell you what it is. And we have a platform that supports target discovery and de-risking and accelerating this, finding the right biology, finding the right targets through the use of virtual cells. And some of you might have already heard of virtual cells. If not, don't worry, that's what we're here to tell you. So we are a virtual biology company, and the aim is actually quite simple at the base. Our aim is to model disease in as close a way to patients as possible, but do it within in silico. And the way we currently look at it is data comes from both preclinical and clinical samples. We turn that into a virtual cell representation, which I'll go a bit deeper into. And we've implemented within our own company a wet lab in the loop. So you're not only learning from the data at the same point and running experiments in silico, you're also using the predictions to generate new data, which again improves the predictive power. So this loop, what we call the learning loop, is incredibly powerful. But what can virtual cells help with? So fundamentally, we build virtual assays, and virtual assays are literal in silico representations of the experiments that you do in the lab itself, and how virtual assays can help optimize what you do in the lab, which assays to pick, which models to pick, and how large a screen to run. In this process, there is data that's obviously available in the public domain. There's data that we generate as Turbine. And also, whenever we work with our partner, they have their own proprietary data, and all three pieces come together beautifully into this virtual cell representation. This we use to, of course, prioritize and position targets, but also at the asset level. So the drugs, the combinations, and of course, the biomarkers, which will help identify where to position the drugs and the combinations. This can be quite flexible because this is in silico, so you can have a small molecule virtual cell, antibody drug conjugate driven virtual assay, and any other modalities down the line. And we'll show you a good example of how we do that within the small molecule space. But fundamentally, we are doing all of this to deliver high confidence preclinical data packages. And those of you on the audience who are from the pharma sector, you will appreciate that having this evidence data packages are critical from the governance standpoint to make sure that we have the right investment going down the line and the right probability of success. And virtual cells, virtual biology platforms are an excellent way of increasing the downstream probability of success. And you will see a couple of use cases about how we do that. As an organization, we have been around for about 10 years, so we're one of the pioneers of virtual cell representation, which you might have heard of and read quite a few very exciting recent papers about. We've implemented our virtual cell platform in more than 30 programs across a host of industry and academia partners, some of which we've highlighted on the slide itself. We are a small company, small to medium size, so we're currently about 90 scientists across AI expertise, biology expertise, and translational expertise. And so far, we've raised just over 60 million euros as an organization. But fundamentally, we are probably the only full stack virtual cell platform. And when I say full stack, what we mean is the entire loop that I was talking to you about before. Where we have not only model thousands of preclinical models, but also implement the lab in the loop system. And what you'll see towards the end of the webinar is we've built a virtual lab. So this is a no-code decision support platform where any one of you could very easily run simulations on a virtual cell. But fundamentally, what is the return on investment for all of this? Why would you want to use it? So number one, it's about 3 to 5 times faster decision-making support. And we've shown that through our own use cases. So there's not a number pulled out of thin air. So we have consistently seen these timelines being delivered. At the same time, when you run experiments, you want to run the most optimal experiments because all of you in the lab are spend a lot of time, effort, blood, and tears, but we want to make sure that the experiments and the time you're running, the resource you're spending, is optimal. So we've shown that using the virtual cell first, using our platform first, and then running an experiment, you can run 50% less experiments, but get 90% of the value and the signals that you usually get. You can identify novel hypotheses, but at the same time, because it's in silico and you're not constrained by the number of models that you can screen, you can run about 3 times more screens. And the scale up is quite significant because you can run millions of these simulations on the virtual cell within a couple of hours. Now we look at our platform, our learning loop in 4 different buckets. Different but highly interconnected. So the first one is the virtual cells, which I already talked about. So this is a scalable AI architecture where it absorbs a lot of the data that from preclinical and clinical sources, and it becomes as close a representation of patient disease as possible. Once you have a virtual cell, we now want to implement a virtual assay. Now, a virtual assay needs to reflect the question that you have. For example, I would like to understand a gene dependency. So I would run a virtual CRISPR. I would want to identify which of my 20 combinations I want to prioritize and then focus on the most promising ones. If I have a pool of 50 targets, I want to rank them, and I want to have an explainable way of ranking these and then defending why top 1 makes more sense as opposed to number 50. And these are all the virtual assays that we can implement within our platform. The third is the virtual lab, and this is a very user-friendly interface where you can select a model, you can select a virtual cell, you can select a virtual assay, and you can select, you can identify your own hypothesis. And this is what you will see from my colleague towards the end of the session. Now, all of this, the glue that holds this together is our lab. So a lab is not only for validating the predictions that come through, but we use our lab to identify what gaps exist within our virtual cell and then build the data sets so that the virtual cell becomes better. So the loop, the more number of times the loop runs, the better the prediction becomes, the better the outcome is, better the more focused and more a higher probability of success of the recommendation that comes out of it. And by implementing this process, through this process, we've identified quite a few lessons, which I'll go through very quickly. The first one is data sparsity. I think for any of such methodologies to work, we need to have good data. And I won't go through all the data types that are mentioned on the slide, but fundamentally, this is a highly multi-omic virtual cell model. So it takes in data from genomics, transcriptomics, a lot of perturbation data coming from CRISPR screens, both monotherapy and combinations, but also viability screens at the same time. And there's an underlying prior biological network that absorbs all this information. And as I was saying before, wherever we do not have the right piece of data to answer the right question, we then partner with external CROs and data providers who are listed on the slide at the bottom to make sure that the virtual cell is capturing the level of data that we would require to answer questions in a very confident way. And what we realized too in this process is, number one, an integrated, diverse piece of data is far more valuable than very large homogeneous data, because from a disease breadth perspective, the more diverse data we absorb, the more confident the model is in terms of prediction. And also, of course, interesting events are rare. And the way we assess any of these AI models with all the AI currently happening, the way we assess if this AI is actually useful is by having very standard benchmarks. And what we've realized is that the benchmarks need to reflect the question and the exact measurement that you would want to use for your question. That's the first lesson. The second one is virtual cells by themselves. They're very interesting as a piece of technology, but they are not a product. What is actually useful from a biological standpoint are the virtual assays themselves. And within the context of Turbine, We use our virtual assays to address three primary questions. The first one is the target discovery and de-risking, which is what this webinar is about. We have an antibody drug conjugate and a small molecule discovery platform as well. And the third one is translational. How do any of the preclinical observations translate into higher models, higher disease models? But we've also learned that there is no general virtual cell. So there is no one bucket where I throw in all data and expect a miracle to come out of it, because biology is complex. And in order to tease out the very specific biological intricacies, we need to have a view at a virtual assay level, which is the exact experiment that we need to replicate. And that is precisely what the virtual assays do from a target discovery perspective, from an ADC perspective, and so on. The virtual lab I've already referred to a couple of times. I won't spend too much time on it because you will see a quick visual, a quick video of it at the end. But the virtual lab is a biology platform. It's not just an AI tool. AI is useful when it can answer a biology question within the context of what we are talking about. And one of the main limitations is in the current scheme of things, where a biologist needs a data scientist or an AI individual to work very closely together in order to implement AI within your system, within your operations, within your workflows. Virtual lab ensures that both the biologist and the data scientist can work very closely together without having a core interdependency on each other. So you can run, simulate, analyze, and recommend within the same context itself. And this ensures that the process is highly seamless. And the lab in the loop, which again, I mentioned a couple of times. So every piece of data that we've generated, either in-house or every piece of data that you generate within your own organizations, your own teams, is highly valuable. And we need to ensure that we are learning from every one of these data points, whether it's a positive data point or a negative data point, does not really matter. So we need to ensure that we are learning from these continuously. And lab in the loop ensures that you're actually doing this. There's a lab in the loop that we've established within Turbine, within our own systems. But the point is that every lab could very easily become a lab in the loop system when you have a platform like a virtual lab. So the virtual lab can inform what the next experiment you should do. You run your experiment, feed in the result into the virtual lab, and then this process keeps repeating till you find an answer to your question. So every lab could become a lab in the loop with the virtual lab itself. Right, coming into the core biological questions the virtual cell will answer. So the first one is, of course, the identifying and de-risking new targets. And the reason we talk about de-risking as opposed to target discovery is having a set of genes being recommended to throw at you is not something we want to do. What we want to do is answer the question, if a new target becomes a drug target tomorrow morning, who is the patient that will benefit from it? What will they combine this with? What's the biomarker that will get the most out of this particular target? So we want to address all those questions. And that's the discovery stage, as we call it. The second one is efficacy. So moving from a biology focus to a pharmacology focus. So modeling in vitro efficacy, which models to pick, what biomarkers to identify, and of course, going into dose-limiting tox as well. So what's the tox profile? What's the tox risk of a given target or a drug? And last but not least, within the translatability piece, which is all these observations which I'm seeing in an in vitro setting or a digital representation or a virtual representation of an in vitro setting, how does that translate into virtual mice, for example, a virtual PDX or a PDO? And this translatability is critical. And from a Turbine standpoint, this is the space where we play, where there's all the way from starting early discovery to translational, making sure that the right data packages are delivered for the clinical stage of any drug discovery and development project. Virtual assays are, as I mentioned, simulated lab experiments. But within the context of target discovery and de-risking, why is it useful? I won't go through each one of this. I think you all have the opportunity to request these slides, and we'll send you these slides. So you can look at what are the questions and the decision points that virtual assays can enable. But the fundamental points which we would like to highlight here is the value of virtual assays for your questions. So the first one is that you can do rapid testing of hypotheses at scale. One of the biggest bottlenecks we have within the industry is we start with, I mean, if you, I'm sure most of you would have heard of widening the drug discovery funnel. So we start with many hypotheses, and at the end, very little comes out. And that's also curtailed by the fact that we do not have the capability or the capacity all the time to test every single hypothesis that we have. But within a virtual cell platform like ours, you can run massive number of hypotheses at scale and reach the hypothesis, the highest confident hypothesis for you to take forward. And there are various decision points you would need for different stages of the target discovery and de-risking pipeline. And we can have a virtual assay, and we have a virtual assay aligned at every one of these decision points. So this could be mechanism of action, it could be validation, it could be connecting the dots to clinical relevance or combination potential. So every stage you can answer with a virtual assay. And of course, it's iterative. So the more number of times you run the loop, the more confident you get, and the more confident you get, more focused is going to be your downstream investment within validation itself. And at the end of the day, what we want to help you all achieve is reach a de-risk target shortlist much faster than what you're currently able to do. And we would like you to do that with a virtual assay platform. I'll introduce the target discovery and de-risking product, which we have, before handing over to Chilla to take you through a very focused use case, a real use case, so that you can appreciate the value of what such a platform can deliver. Now, the aim of target de-risking, discovery and de-risking is to not just identify and prioritize, but also to de-risk targets. And there are multiple ways of doing it. I think one is to model the gene perturbation itself. So what happens when I perturb a particular gene? So I knock it out, and then model what happens downstream. So what does the post-perturbation RNA-seq profile look like? And does that help me identify novel pathways? Once I do identify novel pathways, within this context, can I identify biomarkers of both response or resistance? And would this hypothesis around response and resistance, does that help me identify multimodal combinations? And all of that is connected through a primary question, which we would want to ask is, what is the safety risk of a target which we are recommending? The whole process itself, the framework itself, we have started off with oncology as our primary disease focus, but right now we work across multiple indications. So we've recently extended into immune cells, and we are looking at other neuro indications as well. There are a collection of virtual assays, and this could be both single and double perturbations, partial inhibition, but also some phenotypic assays as well. And we are currently working towards a safety as an endpoint too. So modeling perturbations within healthy cells. What's important with all of this is explaining what happens when I perturb something. And the platform itself will help you go quite deep into the mechanistic explainability of a perturbation, downstream effect of a perturbation. And that is what adds to the de-risking process itself. As I mentioned, this goes across therapeutic areas, this goes across modalities, and we're working with quite a few ADC projects as well. So connecting the immune side with the small molecule side is something we are currently working on quite diligently. And I think with that, I will hand it over to my colleague, Chilla. And in the meanwhile, if you have any questions, please do post it. And at the end, we will address all your questions. Chilla, over to you.
Thank you very much, Krishna. So welcome, everyone. I will walk you through some real-life specific examples just to show you how we are using currently our platform, what outputs we are analyzing to produce those preclinical data packages that are then used to de-risk the target discovery and later potentially the drug development process as well. So as Krishna mentioned, the main output that we are producing is a detailed target data package that contains several important attributes to a target. Let it be a first-in-class target, or it can be also a best-in-class target, to make sure that the most information and the best hypothesis can make their ways into downstream steps and potentially the drug discovery pipeline. So here we are showing an example of how the target data package and the decision target data package looks like. So our virtual lab platform enables running different type of simulations at scale. And as Krishna mentioned, it allows you to test context-dependent gene dependencies across a wide range of biological models, that those be cell lines, PDX, PDO models, or potentially patient samples as well. And you can also test a wide effect range of these perturbations and combination of perturbations, which will then allow you to have viability readout and also, for example, pathway activity, pathway level readout to inform the target mode of action. And I will show you some specific examples and plots for that. These experiments can then inform better mechanistic understanding of target action, comparison of the target MOA across a wide range of very different molecular contexts. Those can also be virtually generated molecular context space, if you wish. And then this mechanistic understanding will give you a hypothesis on how the perturbation and the cell response and then the disease can be linked and can inform predictive biomarker identification for patient stratification. Also, you can explore combination partners for the specific dependency. And for this, from this, you can then design wet lab validation experiments that you can very specifically run to test the target mode of action and the dependency on your specific context. So this is, in a nutshell, what the virtual lab platform and the simulation can provide to this target data package. But we are also linking this information with very important data like cancer relevance of the given perturbation, frequency, for example, of the biomarker candidates within the patient population, assessment of toxicity and therapeutic window that Krishna also mentioned. And this is also on our roadmap to bring that into the virtual experiment space as well. And we are also able to do commercial evaluation and also for best-in-class targets, comparing the target to already available assets to inform the target data package and downstream decisions. And as Krishna mentioned, it is very important to highlight here as well that building this data package is an iterative process. So the virtual lab platform allows us to upgrade the different models that we are using within one project, and then quickly test new hypotheses, quickly add newly generated validation data, for example, and then go deeper, investigating the same research question or build smarter and smarter models to ask new and new research questions on the same target as we are building the package. So This slide is showing how we are actually building these target data packages and how we are utilizing our virtual lab capabilities to assemble all this data and all this information. So at the kickoff stage, we are defining the very specific research question that we would aim to answer using our virtual lab capabilities. And I'm going to show you one short and another more extensive example of such scopes. Then we will set up the specific model to answer that specific research question, so we can go after data within the public domain and also within the proprietary domain. We can also generate very specifically data if we see that there are gaps in the public domain. And then these data will go into training the first version of this specific model that we will use for the first high throughput screening. Within this context, it is usually CRISPR KO screen to identify context-specific genetic dependencies. And we are not only generating a hypothesis and a long target list, but we are also using several in silico generated data, such as effect size, effect strength, and also the different annotations to actually rank the hypothesis that we can generate in the virtual lab to make sure that at the end, with the data package, we can support decisions and bets on the most promising targets. And once we have this target data package, we can also assemble an experimental validation plan. The aim there is to get conclusive validation data based on the simulation data from as low number of wet lab experiments as possible. So we are here using the simulation output for cell line selection. We are using the mechanistic hypothesis to identify PD markers that we can track in the validation experiments. And then these experimental data can also be fed back to the model. So I will show you two examples. One is the NEC1 case study that you can also find in our website. So here we can show that we identify NEC1 as a novel target and the dependency that DepMap could not pick. So at the time when we were running this study, NEC1 was very new, and you can see here that for several cell lines that we actually took to wet lab validation, even with lacking DepMap data, we could validate the context-specific dependency on NEC1 inhibition. This hypothesis also validated in vivo. So here we again used CDX models selected based on the simulation output data. And you can see here that on the middle panel that in the HSC2 cell line, NEC1 KO results in significantly reduced tumor volume in mice. And then this data package, together with the validation of the hypothesis, actually informed next steps downstream. So we've been working on identifying combination partners that can potentially boost the efficacy of the NEC1 inhibition in selected models. In the next example, I will walk you through the end-to-end process of identifying the target, iterating also with the model, and then assembling the target data package. So this project was a novel target identification project. We wanted to find first-in-class targets in very complex biological and molecular background that is high clinical unmet need. So what we did in this project, from left to right, we set up, of course, the model. So we trained the model for this specific scientific question. The major aim there was to have a model that as much represents that specific molecular and biological context as possible. And here we also have a question around benchmarking. So what we are doing is that when we are building these models, we are always measuring how much they are recapitulating known biological phenomena, including known effects of perturbations, known drug effects, known logical connections between perturbations, and so on. And once we have our front-channel model, we are good to go with moving that model into our virtual lab. And as was mentioned previously, actually run the virtual assays and the simulations that will then help us generating hypotheses. So in the first step, we were running this in silico target screen, and we applied two types of perturbations. One, the virtual CRISPR-KO assay for pre-filtering those genetic perturbations that can show context-specific effect. And second, the so-called knockdown screen, with which we are able to actually test different inhibition levels of the same genetic perturbations, which can again help pick context-specific effects or effects that are hidden by the conventional CRISPR screen. Then out of the hypothesis, we filtered out artifacts and then ranked these hypotheses using those ranking criteria that I showed previously. So the main questions to be answered here is which ones are showing specific efficacy in the given molecular background, which gene perturbations are showing good enough effect, strong enough effect that is robust enough to be tested. And also already at this stage, we were taking into consideration the druggability and potential toxicity expression profile data as well, to have a ranked list of target candidates. And what is worth mentioning here is the mechanistic understanding. So after these in silico experiments, when you are perturbing the different genes, you are not only able to get viability data, but we can also get pathway level, pathway activity data, and you can actually compare the different pathway activity patterns in the responder versus non-responder cell populations. And these can give you mechanistic insights on how and why the actual target perturbation is acting within that specific molecular context. And then when we had the top list of the ranked target and target hypothesis, we then forwarded those to the in silico biomarker screen. And the in silico biomarker screen is again allows you to make CRISPR drug modifier like in silico experiments at scale, so at a very wide range of available and simulatable cancer model systems. And also you can test a very wide effect range of your target perturbations to be able to identify predictive biomarkers, potentially synthetic lethal pairs, or also combination target partners. And within this project, I think we ran within these steps more than 300 million experiments. But as we were building the target data packages, we were iterating with the models and kept going back to our virtual lab platform to ask and answer new and new questions. So how does an output look like? On the left side, you can see this target X, which was one of our top target candidates. And you can see here simulated IC50 values within the test set of biological samples that is here, which represent that specific biological context where we wanted to have the specific targets for. And on the right, you can see the cell line panel out of the biological context as control. So we saw in the simulations significantly lower in silico IC50 of this perturbed gene in the cells with this biological context. And we could also compare the pathway activities within these two cell populations. This is represented on the right panel. So again, you can see the two set of cell lines, one within the biological context and then the control group out of the biological context. And you can see on the top that we could actually identify several significantly altered pathways when we perturbed the test cell line group as compared with the control cell line group. So this fed into how we translated the target MOA and the target link to this specific biological context into mechanistic hypothesis. It is also important that we can produce these pathway activity information also at scale, and basically in any combinations of biological samples. So it again gave us a very good and nice tool to map down the hypothesized target mode of action within our biological context of interest. Here I'm showing how all this data then is translated into mechanistic target hypothesis. So you can see here that we could link based on simulation data and then downstream interpretation and translation, the target X perturbation to several different pathways, we were able to identify several differentially functioning genes within these pathways, which we also then used in the wet lab experiments to track the target efficacy and also to prove this mechanistic hypothesis. So on the next slide, I will show you the validation data. Here, we, on one hand, measure cellular viability. You can see we selected panel of cell lines to control and 4 test cell lines based on the simulations. And in the colony formation assay, we could validate that the target is indeed efficient in this given molecular context as compared to the control cell lines. And here within this cell line panel, we also included cell lines where we had no available data and no DepMap data of target X efficacy. On the right side, you can see cell morphology changes, which is important in this context because these morphological changes we expected and we could link to the mechanistic target MOA hypothesis. So you can see here that in a specific sensitive cell line, if you knock down target X with siRNA, you see this cell morphological changes as compared to the SINT treated cells. As I mentioned, we can also use the mechanistic insights to design PD marker experiments in this setup. These are in vitro data. So we selected here the key pathways that we could link to target X mode of action within this specific biological context. And we also selected, based on simulation data, a couple markers that we then measured in vitro, such as histone H3 phosphorylation, gamma H2AX, and so on. And what you can see here is control and test cell lines where we knock down target X with siRNA again, compared with SINT. And we were following how these downstream mechanistic PD markers activity function changes upon target X perturbation. And we could nicely prove the mechanistic hypothesis that we built based on the simulation data. Here, I'm showing how we used the virtual cell and the virtual lab platform for iterations. So also Krishna mentioned that that it is quite a rapid process to iterate, and we can feed new validation data, newly released, or any new publicly available data to fine-tune our models and refine our hypothesis. So this is exactly what we were doing here. Once we had the wet lab validation data, we had a small data set that we could then feed back into the model for new training, but also a set of new questions from our scientists that they wanted to explore using the virtual lab platform. So we took our frontrunner model within this space and fine-tuned it for a next set of in silico biomarker screen that was using new data and all the knowledge that we acquired as we were building and validated the hypothesis. So here again, we used target perturbation, and then for the biomarker screen, a secondary combination perturbation to see which second genetic perturbations will move the target efficacy in either sensitizing or resistance-causing directions. And we translated these findings into predictive biomarker hypothesis. Again, it is important to emphasize here that we also have the workflow and the logic of how we are filtering and ranking this hypothesis. So what was true for the target ranking is also true for the biomarker ranking. We can rank these hypotheses based on effect size and effect strength, statistical significance, potential tractability, prevalence of the biomarker candidates, and so on. And what we were aiming here is to mimic phase 2 precision clinical trial-like setup. So we set up a statistical analysis where we were aiming to rank those biomarkers up which show the highest precision for the target X predictivity. And as you can see on the next slide, we were able to identify such biomarkers in clinically unmet need patient populations. And we also compared the biomarker predictivity to known biomarkers of another target that is already in clinical stage within the same molecular context, and our predictions and our biomarker candidates seem to outperform the predictivity of the known biomarkers of that known target in phase 2. So I can't see the content of this slide now, but it's about the virtual lab. So we built, it's loading now. So we built a virtual lab platform, which is actually a no-code platform that can anyone use to run these virtual experiments and then analyze the data seamlessly and at scale. So we are going to have a short demo video at the end of this presentation, which is going to show you how it looks like and how it works. But the idea is that we implemented the logic, also the workflow of how you can go to target identification, to target derisking, and this logic can help you get to preclinical data packages that can support decision-making downstream. So as a summary, before we go to the video, within this endeavor that I just explained, we aim to find novel targets in a large and biologically complex search space, which represents a clinical unmet need. So we set out to build a model for this and then run virtual assays at scale, knockout, knockdown, CRISPR drug modifier, and other combination screens as well. to identify and then explore at the mechanism level or the target dependencies and efficacies within this context. We could identify and also rank and validate the target hypothesis, and we could also provide a biomarker data package that can inform how we think about positioning of this target candidate at a later stage. Our top target hit and also the simulation-informed MOA hypothesis was validated in vitro, and as the timing, we were showing that we could generate this decision-grade target data package in more than 50% less time as compared to the industry average. So next, we will show you the video of how our virtual lab platform works. And after that, we will have a couple minutes for questions as well.
In this demo, I'll show you how to use Turbine's virtual lab to go from a target discovery question to a decision-ready data package. The goal is to demonstrate how teams can run virtual experiments, interpret results, and decide which targets to take forward. We always start from the dashboard. This is where the team manages experiments, review results, and builds data packages. In a real workflow, this is where collaboration happens. Experiments, analysis, and decisions are all tracked here. From here, we move into experimental setup. The platform is powered by virtual cell models trained on large-scale perturbation data, which allows us to simulate gene perturbations in silico. In this example, we're looking at gastrointestinal cancers and asking which targets are selectively vulnerable in this disease context. First, we define our test group, the cell lines relevant to our indication. Then, we define a control group to compare against. This allows us to identify targets that are specific to the disease rather than general dependencies. Next, we select the gene perturbations we want to test. Typically, a large-scale knockout screen at first. In a live environment, this would run a virtual knockout screen across thousands of genes. For the demo, we open a pre-computed experiment to focus on analysis. The first step is identifying selective vulnerabilities, targets that are essential in the test group, but not in controls. We can then filter and rank candidates based on effect size and prediction confidence. The key difference is that we don't stop at ranking. We can explore pathway level changes to understand the mechanism of action, why a target behaves the way it does. This helps move from a list of hits to a biologically meaningful hypothesis. We can also evaluate which cell lines respond, potential safety signals using tumor versus normal expression, and druggability or novelty. Together, this builds a more complete picture of the target. All of this is captured in a structured data package. This is what teams take into decision meetings, combining ranking, mechanism, and supporting evidence. The goal is to make target decisions more transparent, defensible, and faster. From here, teams can design follow-up experiments, feed new data back into the system, and iterate. This closes the loop between in silico and experimental validation.
Excellent. Thank you, Chilla. And thank you all for joining us. So a couple of points here. One, hope that the use case, but also the background of what a virtual cell is was useful to you. Do reach out to us if you have a hard-to-test hypothesis, you want to benchmark your target, you want to explore a combination strategy, identify good biomarkers, or assess translatability of some of your preclinical projects. Reach out to us because our job primarily is to identify the hardest biological decision-making problems, and we'd love to explore this with you using our virtual biology platform and de-risk it. We have time for one quick question. I think one which actually goes to the confidence question. Chilla, the use case that you showed was around novel target, identifying novel target, but have we quantified how many known and established targets a virtual cell can actually recover? So is there any recall of known targets?
Yes, of course. So when we are benchmarking the model, that is one of the key benchmark metrics, how many known targets we can recall. Yes, we could recall multiple clinically relevant and known targets, also clinically relevant drug actions within this model that we've been using.
And no, thank you for that. And if I recollect correctly, so we did a benchmarking with DepMap, of course, which is the accepted gold standard around here. And we could see that, as in the case with NIC1, which is one of the examples Chilla gave, that we were able to recover more clinically approved drug targets than DepMap did because we model our perturbations slightly differently. The next one is a question which we get asked quite a lot, so I think I'll quickly address it. So is for those who are into technology and AI in the audience, so is the virtual cell in essence a foundation model is what is being asked? It's a good question. The way we look at it is it's a foundation model is a generic framework where you add in a lot of data and then you expect the foundation model to capture biological signals, which it does great. Where a virtual cell goes one step further is it becomes more of a fine-tuned model where you're modeling the perturbation output itself and not just capturing the disease biology. So you could ask more questions of a virtual cell than you would of a foundation model, because virtual cell is far more context specific and it's far more fine-tuned for the data that you have. But yeah, good, useful question. And if you have more questions, please do reach out to us. We are quite open in the way we share our science. So there are two QR codes on the screen that you can see. One where we put all our publications, all our white papers, all our science. That's the first QR code. And the second one is if you liked what you saw and you would like to explore this further, register for early access to the platform itself. We're going to launch at EACR in 10 days' time in Budapest. So if you are attending, please do join us. And thank you all very much for joining us for this webinar, and reach out to us if you have any questions. And thank you, Chilla.
Thank you all.